we explore the cerebellum using optical coherence tomography. The aim of my project was to develop and optimize an imaging protocol to examine the structure of the deep cerebellar nuclei using optical coherence tomography. Just as a nice little warning, I'm most probably going to shorten deep cerebellar nuclei to DCN and optical coherence tomography to OCT, so just holding that in your mind would be very helpful. I broke my project up into sort of 3 little chunks. I first needed to evaluate whether OCT had the penetration depth required to reach the DCN, as it is deep within the cerebellum. Then I sliced the cerebellum up to try and get more structural information out, and then I'm going to compare that with already known data. I then am going to try and pull even more information out by combining what you just heard about before, cross-correlation, with optical clearing, and try and image the diffusion front of the optical clearing solution moving through the tissue. Cool. You might have come across some words you don't really get in my introduction, in my aims, so I'm going to explain exactly what the cerebellum is. Well, the cerebellum is a region in the brain. You've got your brain here, and your cerebellum is this green region here. And what we're interested in particular is this little purple blob here, which is a nuclei deep within the cerebellum. Physiologically, we would want to image from the outside of the brain, just draw a small hole in the skull and image the cerebellum and see right through to the DCN and get all the information out we can. So that's why we started with trying to examine the most physiologically applicable region. So what does the cerebellum do, and why are we interested? We're interested in the cerebellum because it's a nice, simpler model of the brain and is in charge of regulation of movement and motor learning. So when I walk across the stage, my cerebellum is double-checking all of my intended movement against my actual movement. So if I catch a lip of carpet that I didn't expect, my cerebellum will detect an error signal and go, you've not quite done what you intended, and try and correct the motor program so that doesn't happen next time. So I thought I'd explicitly say that I'm not using human brain, I'm using mice brain. There are a lot more of them around, but they are a highly scattering medium. So where OCT was originally developed for ophthalmology and transmission through a transparent medium. And I'll establish what's currently used to get the morphology of cells. Histology is the current practice, and you have to take your tissue, either cool it or solidify it in some way, which would be fixing it, then you section it, and then you stain it. So there's a lot of chemical processing and physical manipulation of the tissue. You can't exactly do that on a living animal. So here's the circuit underlying the cerebellum. And what we're interested in is in the deep cerebellar nuclei, these guys here. And they're interesting because they're the sole output of the cerebellum. So we have the intended motion, which I was talking about before, comes up this side of the circuit and all converges onto this Purkinje cell here. Name's not really important. And any error detection will come up through this circuit here and also converge onto the Purkinje cell. And now for the Purkinje cell to output any information, it all goes through the deep cerebellar nuclei. The problem is very little information is known about it because it's so deep and so small. So MRI has the depth penetration required, but not the resolution to find out what's going on. So that's where OCT comes in. So, exactly. So the whole aim is to try and maintain as much of the network as we can. So if we could use the whole brain, we'd have the entire network intact, and we'd be able to really integrate and see what's going on. Even if we have to take those 1mm slices, it's a lot better than between the 5 micron and the 40 micron that is currently undergoing. So, now what's OCT? Here I have a diagram of spectral domain OCT, which is sort of an intermediary version as to what's currently available. At the heart of OCT is an interferometer. So you get a laser source into the beam splitter, and it's split into a reference arm and a sample arm. In time domain OCT, which was originally developed, this mirror here would move backwards and forwards, and that would control your path length and move you backwards and forwards through your sample. What's depicted here is you can fix the mirror if you break up your signal using a diffraction grating essentially creating a spectrometer and measure the different frequency components of the light all at the same time. So you can measure the entire depth scan by taking a Fourier transform of the information you get from the spectrometer. What our system is actually a swept source OCT. So instead of splitting up the signal into multiple different frequency bins, you can sweep your laser source. So instead of doing it all at the same time, you can sequentially examine each frequency. So we have a swept source laser which is split and goes to the reference arm and the sample arm, which then goes back to the detector and to the computer for analysis. Here's a good way to describe what the methodology is a little bit. We've got our microscope head here, which will scan the tissue. So if you take one depth scan, you get one A line of information. If you then take that scan across in one plane, you then get an image. If you then take a series of images, you get a 3D volume. So we've got translating mirrors that go in both the X and the Y direction, and the Z is the depth scan, and you can get a volume. Okay, now moving on to how we're trying to extend this further with optical clearing and correlation map, which was previously discussed. Okay, optical clearing matches the refractive index, trying to reduce the amount of scattering so you can get deeper within the tissue. So you can see in this image here, you've got more detail in the upper layers than in this image here, but in this image here you can see, with the lights off, more information about deeper in the tissue, whereas this one you can't. So by combining that with correlation mapping, we are hoping to see the diffusion of this clearing moving through the tissue. So a little bit on correlation mapping. What I'm trying to base my technique on is a paper published by Enfield et al. In 2011, and they use this formula here, which you have seen pretty much in the course talk, of discrete normalized cross-correlation with the normalizing component on the bottom, and you've got your image A and your image B, so image A and image B. So when you run this algorithm over these images, you get this image here. So you've got areas of high correlation and low correlation. Where you've got static tissue, the image is essentially going to be the same. So you might move a slight distance, and there'll be a slight time difference. So the fluid inside the blood vessel will move, as this is skin, but the skin will essentially stay in the same place. So when you take the correlation, you get low correlation is light in this case, which you get your blood vessels, and you get, and by creating a structural mask, you just get your blood vessel out, which allows you to just map the microvessels within the skin. So I'm going to try and combine that with the optical clearing and map the diffusion front moving through the tissue, hoping to show more structural detail about the density of the tissue at different areas. Okay, so back to my sort of three methods points. I'm going to evaluate the penetration depth, then I'm going to slice the tissue up to get more structural information, and then I'm going to use this hybrid cross-correlation optical clearing technique to try and get even more structural information. This is my initial results for determining whether we can actually just straight up see the deep cerebellar nuclei without any further processing. And as you can see, well, as I can tell you, there's only about 1 millimeter worth of penetration here, and we now know that the deep cerebellar nuclei are much deeper than that. But what you can see, there's a lot of structural information just off the bat. The way of displaying the information here only gives you the surface. I'm trying to develop a way of making the surface transparent so you can actually see the data underneath the surface, because the data's there, it's just sort of being masked by the outside of the information. And if you compare this to an easily available image, you can see a lot of structural information. You might notice that this image here is a lot fatter than that image here. I haven't squished the brain, it's just an aspect ratio thing. This is 10 millimeters by 7 millimeters, so it's slightly out of aspect ratio. So, what we plan to do from here is to slice the brain up into 1 millimeter sections, slightly cheating on the depth penetration side. So we're going to cut along here, and the deep cerebellar nuclei are going to be deep within here. So a few of these slices are probably not actually going to contain the material that we're after. When we do that, here's an OCT slice, an image, just one image out of a whole stack of 3D images, looking sort of down on the tissue, and here's a histological preparation. This is a Nissl stained, which just makes the cell bodies of the neurons pop out, which is why where you see white is the white matter, and the dark is the grey matter. So you can see similar structure on this. What's great, super exciting, is that these nuclei here are the ones we're after. And you can see there's slight contrast in the same region on that one. So without the staining, without the chemical processing, without the very super thin slices, we're getting structural information about where the neurons are, at least in the brain. We're currently working on developing the cross-correlation code for the solution diffusing through the tissue. So here's a slight preview. This is the time 0 image, and I take further images, actual full 3D scans, a minute after each previous one. So here you've got no clearing solution, here you've got clearing solution on the top, which then diffuses through, but also creates a little bit of noise on the way. We know that we can get much better images than this, which will hopefully improve our cross-correlation results, because you're not really seeing anything in here but noise, which is disappointing, but we can definitely improve on it. This was one of our first results, so hopefully to be able to do a lot better. So just to sum up and link back with my aims, I've developed a protocol for imaging brain tissue, which had not been done before. We are able to, we discovered that the penetration depth was not what we wanted, but it has been done. And we then sliced it up and found lots of structural information, which is really promising and really great information. And we now eliminated the need for costly and time-consuming processing. You just slice them up and away you go. And there's lots of clinical and physiological applications. We can now examine larger networks, hopefully get some sort of functional twist on whether, let's say, we could image whether the cells are active or not. That would tell us what cells are active, and hopefully in a time scale that you can see just different regions of the cerebellum lighting up, so you can actually see what's functionally going on. Where I'm going to move on to over the next week or two is to reconstruct those 1mm slices digitally. So we started with the whole cerebellum, we slice it up, then we're going to try and put the cerebellum back together digitally. And then we're going to continue working on the correlation mapping algorithm, move it into the Fourier domain. It should be easier, that's why it was developed, and try filtering the images more to get a cleaner image so we're not imaging so much noise. Then I'm going to repeat my optical clearing experiment from the start, trying to reduce the amount of noise in the original images, and instead of taking entire 3D images, just take a 2D slice through and record it at about 20 frames per second, because I'm suspecting that the diffusion is happening a lot faster than we're capturing. We need sort of sub-second resolution rather than minute resolution. Cool. I'd like to thank my supervisors Igor and Ruth, and both the departments for putting up with me, and both my lab groups for keeping me sane.